Okay, we are going to be moving on to our first biomolecule that we're going to talk about in depth, that is nucleic acids. You guys have probably heard of these before. We're going to talk about it in depth. This is DNA, RNA. I have a picture down here that should clue you in. It's a very common picture now. Something you've seen before, I'm sure. Oops. So, the function of DNA. DNA stores information. This is our genetic material. In fact, I'm going to put over here, this is DNA. Stores our information and has our genes are contained in our DNA. There's a blueprint for building proteins. In fact, DNA is just instructions for proteins, right? And this is kind of the general process, which we will talk about this in much more in depth than we will right now. But DNA, right, gets transcribed into RNA, which gets translated into proteins. Also, the transfer of information, that's where that RNA comes in, right? RNA transfers information to other parts of the cells. This is a blueprint for new cells being formed, a blueprint for the next generation of cells. This is how who started, we all started as one little cell here with our nucleus filled with DNA, and we are now trillions of cells due to this process of replication, right? Which is part of how our cells divide. It's DNA and RNA. So let's start talking about the structures. We talked about ribonucleic acid a little bit. This functions in transfer. It is a single helix, not a double helix. We'll talk maybe a little bit more in depth about that a little bit later. DNA is a double helix. You'll notice that the names of these are a little bit different. Ribonucleic acid and deoxyribonucleic acid. Again, that's something we're going to talk about in the last video in this unit, so don't worry too much about that now. The general structure of DNA, the monomer for DNA, again, monomer simply means kind of a single unit, which makes up these long strands of DNA, is called a nucleotide. Again, this is probably a review for you, but let's take a look at a nucleotide because it has, different parts that are very important, parts that you need to know about. There is the phosphate group. Clearly circled, but you'll notice that functional group over here. This is a phosphate group connected to a sugar over here, a pentose sugar, a 5-carbon sugar. Right. The third part of a nucleotide is the nitrogenous base. These are the three components. You need to know the three components of a nucleotide. That is something that is very important. Three components. I'll even write them again. They're so important. Phosphate group. Sugar. And a nitrogenous base. Okay, so when you talk about DNA, and we'll talk about this more in depth a little bit. Let me see where we're at. If you try to transfer this image over here, you can kind of see that the nitrogenous base is what we typically call the A's and T's and C's and G's, the adenine, thymine, guanine, cytosine. That is typically this nitrogenous base area that's doing the actual bonding. Now this phosphate group over here and this sugar, these are part of the backbone. This is the backbone. That's why it's called a sugar phosphate backbone. Sugar phosphate backbone. So phosphate and sugar make up the backbone, and the nitrogenous base is the hydrogen bonding and has the actual code, right? The code is actually carried in that nitrogenous base. Okay, so base pairing of DNA. You have these things called purines, which you've heard about before, but probably never knew that they were called purines. Adenine and guanine are considered purines, and the way I always remember that is pure silver. Pure silver, because the chemical, we would abbreviate this chemical as Ag. Right? Silver, pure, Ag, pure silver. Adenine and guanine, those are your purines. Your pyrimidines, this is the other one. Thymine and cytosine. Also, I'll mention this here, uracil, which is only an RNA, uracil, which is U, right? So the way I remember that is I remember the pyramids. Pyramids look like they're cut to me. C-U-T, it's like they're cut out of something, they're very triangular, right? So that's how I remember these. Pyramids cut, pure silver, pure Ag. Pairing. So A bonds to T, and there are two bonds between them. So let's look up here at this kind of structure. Adenine and thymine, these two bond together, right? You'll notice that there are two bonds that are forming here. Those are hydrogen bonds. These are hydrogen bonds. Okay. Let's look at C and G. I want you to pause and you tell me between guanine and cytosine, how many bonds do we have holding those together? Okay, you should have looked on this diagram over here and noticed that you have hydrogen bonds over here. And there are three of them. 3 hydrogen bonds between G and C, 2 between A and T. So you might think, well, that's not too important, right? But what that means is that between C and G, you have a stronger bond on your DNA than you do between A and T, which is a little bit weaker because it's not bonded as strongly with that third bond. Something to keep in mind for later in the class. You don't need to know the general structures of purines and pyrimidines, but I do think it's interesting to note that purines are a double ring structure, as you'll notice. Again, remember that there's carbons all over the place in here. Wherever you don't see stuff, there's carbon. And your pyrimidines are, these single ring structures. So let's go ahead and move on. Another thing we need to know about DNA is they are anti-parallel. Now, what do I mean by anti-parallel? Well, you have a 3' end and a 5' end. Would you say that these two strands are, again, here we get A and T bonding, G and C bonding, are these two strands parallel? Sure, they run parallel to each other, but they are called anti-parallel because the other side is kind of runs the opposite direction, right? It's kind of like a street. In a street, you have maybe traffic going both ways, right? That's how I think of it. Anti-parallel. Now you might wonder to yourself, how come it's called 5'? How come this is called 3'? And it's because of carbon numbering. If you were to number these carbons in the sugar, 1, 2, 3, 4, and 5, you'll notice that phosphate group comes off of this 5 carbon right here. And that's why this is 5' end. And the other end of this, on this backbone, is this hydroxyl group down here, right? Remember? Hydroxyl. And this is, of course, phosphate. Two of our functional groups that I said were going to be important. Okay. So this hydroxyl group on the other side over here is on this 3' end. And of course, you'll notice this other image over here is upside down, right? It's coming in a different direction, right? It's coming in a different direction from this side. That's what it means by anti-parallel. It's important to note the type of bonding that takes place. You can only add bases to the 3' end of a growing strand. So the next nucleotide that would be added would come here with its 3 phosphate groups, which we'll talk about a little bit later how that works when we get to replication, but stuff is only added to this 3' end. And when that bond is formed, this bond right here, right? This bond to the next whole thing here. This is a phosphodiester bond. This is a phosphodiester bond. That's the name of it. That's something I would know. I would know what a phosphodiester bond is and where you find it. These are found in nucleic acids. And simply describes the specific bond between two nucleotides in the backbone. Okay. So let's do a little summary here. The monomer for in summary, we are talking about nucleic acids. The monomer for nucleic acids are known as nucleotides. We have pyrimidines, which are cytosine, thymine, and uracil. Uracil is found in RNA only, right? Uracil is RNA only. Purines, adenine and guanine, A and G, CUT. If you remember, A bonds to T, C bonds to G in DNA, right? Of course, also U. Some people remember that as apple in the tree, car in the garage, if that helps you. apple in the tree, car in the garage. A bonds to T, C bonds to G. Of course, use this just for RNA only. Takes the place of T. The polymer for nucleotides, or for nucleic acids, is known as DNA, and also RNA, deoxyribonucleic acid and ribonucleic acid. Okay, the types of bonds formed in nucleic acids. You have a phosphodiester bond, which is in the backbone. Remember, this is between that. This is between that phosphate group and the hydroxyl on the sugar phosphate backbone. And you have hydrogen bonding between the bases. These are kind of the rungs of the ladder for DNA. We talk mostly about DNA today, but that's because RNA is at a much lower stage. We're going to talk about that one day. Function. Nucleic acids store information, and nucleic acids transfer information to different parts of the cell. There's also some other functions of nucleic acids, which I've kind of hidden for you for the time being. We'll talk about that later, but I'll give you a hint. It involves ATP, which is a nucleoside, rather. And we'll talk about that in a later lecture, so don't worry too much about that now. Anyways, this is pretty much all you need to know about nucleic acids. I hope that helped. Okay.